Welcome back. I hope your midterm exam went well. We're going to jump in now with chapter 8, which is cell division.
All right, so before we can actually dive into cellular division, we have to learn a little bit about DNA and how DNA is packed. And the reason for this is because when we start seeing cells divide, we actually are going to be following the DNA in the cell as kind of the major thing that we follow, because that's one of the most important parts of the cells, it's DNA. And so here I've drawn a picture of our structure of DNA. So here you have your DNA, linear DNA, and then your double helix is how we see it a lot. And then what happens is we have these proteins that we're going to end up wrapping the DNA around. So these green things here, these are our proteins. And these are called histones. And the idea here is we want to pack our DNA tightly, because all of our DNA, if we were to take one of our cells and take all the DNA out of that cell and then stretch it out, it would roughly be the height that you are. So roughly somewhere between 5.5 to 6 feet. Now, you can imagine now we have to fit that all in one tiny, tiny, tiny cell. And so if we have it in this form right here, this very loose form, it's not going to all fit inside the cell. But if we start to pack the DNA, and in this form we call chromatin, and this is what we call lightly packed DNA, chromatin. So those are lightly packed DNA, and this is kind of the state that our DNA is in. And that's what we do, is we take the DNA, we wrap it around these proteins called histones, and these proteins, so kind of think of it almost like a ball of yarn, where if you were to take that ball of yarn and spread it out, it's a whole lot. But if you take it and roll it up really nicely, you can fit in this nice condensed ball. And so that's what we do. We wrap it around and we create this chromatin. And then if we need to completely pack it up, so tightly pack it, we then call it a chromosome. All right, so you first go here to our lightly packed, and then you get to your chromosome part right here. And this is tightly packed. And a lot of times we'll draw this so it looks kind of like a bowling pin. So this would be our chromosome. This is eventually what our DNA will be tightly packed up as a chromosome. And when we do cell division, we want our DNA all nicely packed up so that we can then split it and spread it and separate it into two different cells. And we do that nicely as a tightly packed chromosome. So really in your cells inside the nucleus, your DNA is mainly found in this chromatin fate where it's lightly packed. And the reason why we want lightly packed is because anytime we need to access a part of the DNA, we want to be able to go in, access that part, and then pack it back up. If it's tightly packed, then that makes it really difficult to get to. So really, it only becomes a chromosome and is tightly packed when cell division is occurring. All right, so when we talk about chromosomes, there's a certain amount of chromosomes that are in all of our cells. And we have these what we call 23 paired chromosomes, which really means is that we have 46 total. So kind of almost think of these like socks. If you have 23 pairs of socks, you actually have a total of individual socks, you have 46 of them. And we say 23 paired chromosomes is because when we look at the chromosomes or the DNA that makes us who we are, we got that DNA from our parents, right? From our mom and dad. And we're going to talk about that at the end of the chapter, how that kind of works. And so what we do is we get 23 of them from mom and 23 from dad. And so that's these 23 paired chromosomes. That's why we have a total of 46. So once about going back to that sock analogy, let's say if your parents gave you a gift and it was a gift of socks, what a fabulous gift to receive from your parents. One of those socks would come from your mom and the other sock would come from your dad. And so it's the same sock. They look very similar, all that kind of stuff. But one came from your mom, one came from your dad. That's how your 23 paired chromosomes are. One came from your mom, one came from your dad. And we label our chromosomes 1 to 22, and then our 23rd set of chromosomes are called our sex chromosomes. Those are the X and the Y's, and we'll definitely chat a little bit more about those. So when we think of our chromosome, so let's say this is chromosome number 1. All right, and chromosome number 1, we'll call it 1M. This means you received this from mom, and 1D, so you received that one from your dad. Now, if we were to look at the genes that are on this chromosome, they're the same. So in other words, let's say on chromosome 1, now this isn't for sure, I don't know off the top of my head if this is where you found this, but let's say on chromosome number 1, you have the gene for hair color. And that gene is found right here on the chromosome or within this part of your DNA. So on chromosome 1M, you have your hair color, and then chromosome 1D, you would also have a gene right there for hair color. And let's say right next to it, you have the gene for eye color. And so what we noticed from chromosome number 1 that you got from your mom and chromosome number 1 you got from your dad, it has the same genes on there that code for different aspects of who you are. All right? So in other words, you're not going to see height gene here and then no height gene here. You're also going to be, it's always going to be the same. We call this homologous chromosomes. All right, so homo meaning the same. So homologous chromosomes, these are the same type of chromosome. They code for the same types of traits or same characters. All right, but actually what the code is could be a little bit different. So in other words, let's say your mom has brown hair and brown eyes. So even though these code for hair color and eye color, the specific code, so the A, T, C's, and G's, those adenine, thymines, guanines, and cytosines, will code for the specific trait or the color here, so brown and brown. Where let's say your dad had blonde hair and blue eyes. And so even though these are still homologous chromosomes, because they code for the same types of genes, but the specific gene could be different. The specific sequence of the DNA and what it codes for the trait, so the blue eyes, the blonde hair, will be different. So these are homologous, but they can be actual different traits inside them. but they're always the same genes that are coded in there, but the actual gene that it's coded for could be different based off of the trait that you get. So that's how you are uniquely who you are, because you get some traits from your mom and some traits from your dad. So once again, these are homologous chromosomes, which means they have the same gene on there. Now there's another type of chromosome. So let's say I take chromosome 1M and I want to make a copy of it. Oh, I did that wrong. When we make a copy of a chromosome, we keep them together. So we still have these two individual ones here, but they're connected together right here. But if we were to look here, so this is chromosome 1 from mom. So we have brown hair and brown eyes. Since we're making an exact copy, we will see here that we also have brown hair and brown eyes. So when we have two chromosomes that are exactly the same, which means not only are their genes the same, but the specific gene sequence and what those genes code for, like their trait, their hair color, their color, are the same, we call these sister chromosomes. So these are exact copies. So we could do the same thing with chromosome 1 from dad. We can make an exact copy of it. So this is chromosome number 1 from dad. We're making an exact copy. Here we see that there is blonde hair and blue eyes. And because we're making an exact copy, we have blonde hair and blue eyes there. So these here are homologous chromosomes because they code for the same type of genes. These here are sister chromosomes because they're the exact copy of one of those chromosomes. So these are kind of important. I know they're very similar in sounding and what they mean, but these are important for you to understand when it comes to cell division. And so once again, homologous chromosomes are when you get chromosome 1 for mom, chromosome 2 for dad. They both have the same type of genes in there, but the actual trait for those genes could be different. Where sister chromosomes is where we take one of these and we make an exact copy of it. And the reason we're going to make an exact copy of it, because when you have one cell and you're going to divide it into two cells, you want to have all the genetic material shared between the two cells. And so what you end up doing is you make an exact copy of all your chromosomes. So all your 23 pairs, you're going to make an exact copy of them. So you're going to go from 46 then to 92 once you make the copies. And then you'll split those 92 into the two different cells. So at the very end, you end up with 46 again. So each new cell has 23 paired chromosomes. So you make an exact copy of that when you do your cell division. All right, so let me get rid of this right here. And we're going to do a quick breakdown of the cell cycle. All right, so we're gonna have our cell cycle. So this is the life cycle of a cell. So when it goes from a single cell and then divides into two cells. Now, when this is the beginning or end of it, so at this point, your cell has 23 paired chromosomes, or a total of 46. So this is just shortly after it just divided. So we're starting a brand new cell. So in this part here, we have what we call our G1 phase. So our G1 phase, the G stands for either growth or gap, depending on which biology book you have. But pretty much what happens in the G1 phase is the cell grows, because right, you just split a cell into two, so it's a little bit smaller. So it's going to grow, it's going to build more mitochondria, it's going to build out its ER, all those Golgi apparatus, all that kind of stuff, and build. So at the end of G1 phase, we still have our 23 paired, which is a total of 46. Then we have what we call our S phase. S phase, the S stands for synthesis. If you don't know what the word synthesis means to make or to create or to build. And so in our S phase specifically, what we're doing is we're creating more DNA. And so what we're going to do is we're going to take each one of our homologous chromosomes, each one of those, and make a sister chromosome out of each one of those. So our total of 46 chromosomes, we're going to end up creating 46 sister chromosomes. So this is DNA synthesis. So at the end of that phase, we end up with these sister chromosomes, and we end up with 46 sister chromosomes, which is a total of 96 chromosomes, because each one of these sister chromosomes has 2 chromosomes in them. So all we did is of all of our 23 paired chromosomes, we made an exact copy and created these sister chromosomes. Then we have what we call our G2 stage. So once again, G2 either stands for growth or gap. So you could say growth because, all right, we're going to split this cell into 2, so we want to make sure there's at least enough room or enough stuff available to be able to create 2 cells. And at the end of our growth 2 stage, we still have our 46 sister chromosomes, so SC for sister chromosomes, which is a total of 96 altogether. Then we get into what we call our M phase. So our M phase stands for mitotic phase, or the process of meiosis. And then meiosis is broken up into 4 individual phases, which is what we're going to end up talking next once we're done talking here about the cell cycle. At the end of our 4 phases of mitosis, what we end up having is we have 2 new cells, and so what we do is we split all of our sister chromosomes up so that we then end up with 2 new cells, each with 23 paired chromosomes, and each with a total of 46 chromosomes. And then each one of these cells will then go through the cell cycle again and divide again and go through that whole process. Now, when we break this up, we call the G1, S, and G2 phase, we call this interphase. Or pretty much what it is, just the regular life of the cell. And then the M phase, which is still the mitotic phase, right? Mitosis. Now, because this kind of looks like a char graph, you might see that in interphase, the cell is roughly 75% of its life, and mitosis is 25%, but that's not the case. Cells spend interphase of roughly 90% of their life is in interphase, and only 10% is actually in metaphase when you're splitting the cell into two, going through that whole process. All right, so that's the cell cycle. That's mitosis, which we'll dive into here more. And this is how our chromosomes, we copy them, separate them, put them into different cells, and then each one of those go into their relative cell cycle after that and go through the same process once again. Now that we know how the cell cycle works, we're now going to jump right into mitosis. And our definition for mitosis is the division of a somatic cell into two genetically identical daughter cells. So let's break this down here really quick. So first we need to know, what is a somatic cell? So a somatic cell is literally any cell in your body except for your sex cells. And your sex cells are going to go through meiosis or meiosis, which we're going to talk about later. So the division of a somatic cell, so pretty much any cell in your body except for sex cells. So that could be your toe, that could be your heart, that could be your lung, that could be your hair, that could be your eyes, that could be your skin. So any of those cells, those are all somatic cells. So division of a somatic cell, so a single somatic cell, into two genetically identical daughter cells. So we're going to go from one cell to two cells, and when we do that, the genetics or the DNA of the original somatic cell, the two daughter cells that we create from this will be genetically identical, exactly the same. We won't be able to tell a difference between them if we look just at their genetics. So that's what mitosis is the division of a somatic cell into two genetically identical daughter cells. All right, when we looked at our cell cycle, we said in the M phase, or our mitotic phase, or mitosis, we had 4 additional phases. And so we're going to look at each of these 4 phases and what happens in them. So our first phase is what we call prophase. And once again, when I talked about the reason why we need to know about genetics is we're going to actually be following the actual genes inside our cells as we divide them here. So one of the first things that happens in prophase is that we start to break down the nuclear envelope. Now, our nuclear envelope is the membrane around the nucleus. We call that the nuclear envelope. So here's our nucleus, and I'm just going to show it with some gaps in it. That's us breaking down our nuclear envelope. Then inside our nucleus, our DNA is going to go from our lightly packed, so that we showed earlier, which is our chromatin, to our tightly packed, which is our chromosome. So DNA goes from chromatin to chromosome. So from lightly packed to tightly packed. And we show this by kind of drawing these worm-like structures. And actually, when you look at, so usually I teach a lab with this, but because of COVID-19, we're not doing our lab this time. But when you actually look into a microscope, you can actually see the DNA in these chromosome form. You can't see DNA when it's in its chromatin form because it's not dense enough to pick up on a microscope. But when it becomes tightly packed, then it's in its chromosome form, and you can then see the chromosomes. And so they kind of look like these worm structures inside of that nucleus. All right, and then the last thing we have is our centrosomes. Let's actually make these guys a different color. So our centrosomes migrate to opposite ends of the cell. So they originally were up here, so you're going to have one here and here. So these are our centrosomes. All right, so that's prophase. So those three things happen. We break down our nuclear envelope. Our DNA goes from a lightly packed state we call chromatin to a tightly packed state we call chromosomes. And our centrosomes migrate to opposite ends of the cell. And we're going to see how important those are later. So after prophase, our next phase is anaphase. Oh, sorry, not anaphase, metaphase. And in metaphase, what happens is our chromosomes line up in the middle of the cell. So our sister chromosomes, right? So these are X's. So remember we drew our sister chromosomes. They look like this. So here we have these X's represent sister chromosomes. And then our centrosomes are going to attach these protein spindles to our sister chromosomes. So our sister chromosomes are SC. So that's metaphase. Once again, metaphase, we line up all of our sister chromosomes right down the middle. So remember, there's actually 46 of these. I'm not going to draw all of them because that would take forever and make this look very convoluted. But imagine you have 46 of these chromosomes all lined up. Then our centrosomes here are going to attach these spindles, kind of think of them like Spider-Man, just shooting out these webs here to attach to our chromosomes, our sister chromosomes. Then our next phase is anaphase. So in our anaphase, what happens is our centrosomes start to reel in those protein spindles, and that breaks apart our sister chromosomes, so it breaks them right in half. So once again, here's our sister chromosome. And it's going to break them in half and have one go this way and the other one go that way. All right, so our centrosome pulls apart our sister chromosomes, pulls them apart, and then reels them to the opposite sides of the cell. So now these are heading to opposite sides of the cell. All right, so that's our anaphase. And then our last phase is called telophase. Now, our telophase, our last phase, we're going to be reversing some of the things that happened down in our prophase. So the first thing we're going to do is build back up our nuclear envelope. And then our chromosomes are going to go back to chromatin, so from tightly packed to lightly packed. And then lastly, we're going to start to pinch off and split our cell into two. And we call this cytokinesis. So this pinching off here in the middle, and then what we end up having now is our two new cells, and each one of them have 23 paired chromosomes with a total of 46. Since we split up all of our sister chromosomes. And once again, they are now genetically identical daughter cells. Why are they genetically identical? Well, remember, when we create a sister chromosome, we're making an exact copy of that other chromosome. So in other words, we have 2 exact copies of every single one of our 23 paired chromosomes. And all we did through mitosis is pack them up, line them up, split them up, and then divide the cell. That is pretty much how mitosis works. And so really the way I always remember this is when I'm thinking about metaphase, I know that I'm lining up my chromosomes down the middle. So metaphase down the middle. So MM, middle, middle. When I'm doing anaphase, I know that I'm tearing my DNA or my chromosomes apart. So anaphase apart, AA. Telophase, I'm dividing my cells into 2. So telophase 2, dividing them into 2. That's how I remember which of these phases come after prophase. So all you have to do is remember prophase come first, and then you can always figure out, okay, what's the next phases and what happens in those phases by thinking, okay, metaphase, line down the middle. Anaphase, apart, we split them apart. Telophase, 2, we create 2 new cells, and we split the cells into 2. All right, that, my friends, is mitosis. Next, we're going to look at what happens when we do meiosis or meiosis, however you want to pronounce it.